one original copy. And the good thing is you can then easily spot sequencing errors. If the copies don't agree, that must be a sequencing errors. But if here's a real change from A to C to A, you see that in all the copies, you can be relatively sure that it's a true change. And in order to get enough copies of each original molecule and to get deep data to identify 1 in 5,000 mutations, we need to go to coverages that go way beyond 100,000x. So much higher than you would normally go for a human genome, for instance, where you take maybe 30x. So that's why I call that ultra deep methods. You can then group these and check how well you can do error correction. So here we termed that correction level, meaning that if I find 3 copies of an original molecule, I have a correction level of 3. If I find 4 copies, I have 4. And as you can see here, with 1 copy, so no correction, the error rates are relatively high. They are slightly different for each nucleotide change. And with 4 copies, you basically reach an error rate that is negligible, like 1 in 40,000. So now we can actually detect variants with 1 in 5,000 or better. One problem we had to face is that we had to optimize the PCR cycles and all that stuff because, well, it's a kind of a random process how many copies you find for each of the molecules. And our optimum lies here around 3 or 4. So we had to optimize the protocols to get as little as possible coverage of the 1x and 2x. But we didn't manage, as you see, to get that perfect. So about half of our reads will not have enough copies to get the optimal error rate. And the other problem is if we have many more than 4 copies, then we lose sequencing power. So we don't want many more. So our optimum would be to have a peak at 4 copies. But that's really tricky and doesn't always work perfectly. So finally, coming back to our graphics, we now have the error rates to really detect something at 0.2% or 0.1% fraction in the tumor mutation in the plasma. And now we could develop different algorithms. For instance, when following here a simple graphic of having only 4 target mutations in 4 oncogenes, we can develop models that follow the curve over time and predict if a patient is in remission or progress or stable. And we can also make methods that check at the end of treatment if the patient still has a tumor load. So we call that minimal residual disease. Or we could also continue to track the patient with liquid biopsy and see when there's a relapse. So many methods possible. And that's how that looks in real life. Normally, as I said, we track 30 or more mutations. So each line here is 1 mutation in 1 patient. And that patient was lucky. So basically tumor-free after the treatment. And here is a worst case where there was an initial improvement, but then there was clearly at a later time point, so each of these are time points, a progress of the patient. You can also see that the SNPs are kind of following the same trajectory. They do not have the same original level because already in the tumor itself, they might not have the same fraction of cells affected. So there could be SNPs that are affecting less cells of the tumor than other. I don't want to go deeper into that because I want to come to our application of that exact same technology to virus sequencing. You all know these screenshots from Next Strain, for instance, application, which follows the evolution of SARS-CoV-2. And yeah, you can see Omicron here and Delta. And when we started that project, it was here in May 22, where there were much less variants. And many people thought that SARS-CoV-2 is quite stable, actually. It has very little variants, but we already saw the first variants coming up. And we were wondering, is there also variation within one patient, or do patients normally have one clonal population of virus variants? So we wanted to, similar to cancer, find variants that maybe affect only 5% of the viruses in one patient. So we set up comparison benchmark setting, we took the standard Arctic panel at the time, or still the most commonly used Amplicon panel. There were other Amplicon panels like Eden, and we wanted to compare that to a method that's close to our liquid biopsy, where we use oligo hybridization, so NGS panels that use oligos to enrich the virus from the sample spit or whatever sample you took. And we tested two technologies for that, IDT and Twist. IDT, you might have, Twist, you might have worked with when you did exome sequencing or any other type of enrichment. They have a very efficient oligo-based enrichment technology, and they, in 2020, already released a specific kit for enrichment of the COVID-19 virus. So that was very useful. The problem was in the beginning, they didn't have UMIs included. So we decided to test another technology, IDT, which had a protocol at the time called XGen Prism. They now renamed it to XGen cfDNA, but it works perfectly fine for viruses, not just for circulating cell-free DNA in plasma. And that one directly includes the UMIs in the protocol. So you attach them while you add your adapters. And what was even better, the problem was IDT did not offer a SARS-CoV-2 panel. So you had to either design these oligos yourself, or you could combine it with actually the Twist oligos and fuse the protocols. That's what we originally did. So now we had a way of enriching the virus, and we could add UMIs. We were very happy in the beginning that we saw that the coverage distribution of oligo enrichment is much more stable, so smooth, compared to these Arctic protocols. Arctic is often run with Illumina or also with ONT, with nanopore, but you can see they are really spiky, and here in the oligo enrichment, they are much more smooth. The only problem with oligos is when the CT values become very high, like 30 or higher, you don't get coverage at all, so the enrichment doesn't work anymore. So finally, we can do the same as in liquid biopsy. We have our virus population, and we ask if there is a small fraction in the host, so in the same sample, that carries a novel mutation. And we want to do ultra deep sequencing because the virus is so small, 30 kb. We can even go to up to 1 million X coverage, and otherwise want to do exactly the same as for the liquid biopsy. The problem was that, well, at the time, we didn't know if there are actually subclonal mutations. And even if there are, we had no gold standard. If we took it from a patient, a sample, we didn't know if there are any that we can use for benchmarking. That's different from cancer, where we already know from the tumor that a specific mutation should be found. So what we decided was to go back to Twist again and order their synthetic viral controls. By now, they have probably like 30 different synthetic viruses for SARS-CoV-2, and they can be ordered. They are similar to Delta or Omicron or to the original Wuhan and so on. So with that, we could now make synthetic mixes. So we could take a control that had almost no variants, very original, Wuhan-like, and we could then take, for instance, Delta or Omicron and so on, and mix the one with more mutations into the one with less. And we could modulate any sort of mixture. And we did that down to 0.1% actually, to check how sensitive ultra deep sequencing can be. First thing, we were happy again that the coverage in these synthetic samples looked quite similar to patient samples. So if we use 1 million copies of the synthetic virus, we got it was comparable to a CT value of 15, and 1,000 copies comparable to a CT value of like 27 or so, 28. So that's good. We can even modulate the sample, so we can simulate samples of different quality, and we can simulate different fraction of viral mutations. We use unique molecular barcodes again to allow to be really highly sensitive by having almost no errors. So that's pretty much the same protocol as we used before. And we do see exactly the same error correction. So it's very comparable to the tumor, to the plasma data. At 4 copies, we see extremely good error correction that allows us to call 1 in 10,000 or less mutations per molecule. So if you have only 1 virus per 10,000 viruses in a host that has a mutation, we can still detect that with that error rate. Now we have to come to the statistical model. We developed a tool called UMIVAR. We already developed that for cancer, but it works also for viruses, where you look at a specific position, the coverage. Let's assume we have 1000x coverage and we have only 3 reads that show a difference, a C instead of a T. Can we rely on that? And that we can modulate or model by negative beta binomial error distribution, where you can see that if you have no error correction, you still see quite a lot of positions that have 4 reads that are different from the reference. Here, like 4%, or 3, you have 12%. So with that, we could not call that as a variant, because we would get 12% of the viral genome would be called as a variant, as a mutation, which cannot be true. If you go to 4x correction, you can see that this is not the case anymore. All these positions that have 3 or 4 or more errors, they disappear. So when you see in that correction level a variant that only shows up in 3 out of 1000 reads, you can already trust it. It is very likely a true variant. So we would again try to reach 3 or 4x level. We can still use these reads because our model will automatically recognize them as more error prone and will automatically require more changed reads. All of that we put into a pipeline and we compare to other tools that can achieve the same, like IVAR is pretty good, and some older tools, VARScan and LowFrec. All of them can detect low frequency variants in the Illumina data, and then we can compare them. And what we found is that this works as well as in cancer. So here, just 4 variants from one of the strains we mixed. And you can see that in the 1% mix, we see roughly 1% of the reads changed. So 0.9%, 1.05%, so very close. For the 0.5, it's equally close, although there is a bit more variability already. And then for the 0.1% mix, we were a bit too high. We don't know if that's a prediction problem or if it's the mix itself. It seems like might have been a bit higher than 0.1%. But in general, we get extremely close. So we could check that in many different mixes. So for instance, here we did a mix of 50% and 100% and 0. And we see that we can perfectly observe what we expect. And we see that for different variants and for different samples. And I already showed now the mixtures where we had much lower, so only 0.1%, 0.5%, and here 1%. So this one here is 1 in 1000. And we still get a very close call in our observed reads. So that method works very well. And thanks to the UMIs, it's highly accurate because you can also remove any type of PCR duplicate problems that mess up statistics. In real data, it looks a bit more messy. Here we have real patients. You can see here our p-value of the predicted variant and then the allele frequency. So higher p-values are better. So up here is the true stuff. And you can see, we see the expected clonal mutations at 1%. So these are the ones the whole population is affected. But we also surprisingly see some variants at around 40 to 60%. So some patients seem to have at mixed populations. And then we see a lot of low frequency variants. And that's where we still are working on. It's still a bit too high. In our opinion, we still optimize our tool, get better p-value thresholds to really come up with these few low frequency variants that are likely true. So that's my conclusion for that part. And I'm jumping now to the second part, but I would say that ultra-deep sequencing combined with unique molecular barcodes is really powerful, and you can achieve really amazing stuff like finding a mutation in 0.1% of your sample. Now, as in the title, I promised to have a second topic, which is ultra-deep microbiomes with nanopore sequencing. So this ultra-deep sequencing is equally interesting for nanopore. Many of you have probably worked with hospital isolates or generally bacterial isolates. In my case, I use them to detect antibiotic resistance genes in the bugs we collect daily in the hygiene department. And one of the biggest problems are antibiotic resistance genes on mobile elements, on plasmids. And there we use nanopore, because with nanopore or also PacBio, we use both. You can really get very long reads that allow you to perfectly assemble these chromosomes and plasmids in circular chromosomes or plasmids, so you can be sure it's a plasmid or not. And by that, then by annotating the resistance genes, we really understand how a specific resistance or extreme resistance spreads in the hospital. The problem is we want to do that with microbiomes so that we don't have to make the isolates anymore. But with microbiomes, when you assemble, you still get this mess of an assembly graph, especially if you did low coverage microbiomes. So can we fix that with ultra-deep sequencing? It was a project I long wanted to do, but well, as it happens, another project came first because of a good collaborator in Tübingen, and that's natural product genome mining. So in that case, you use bacterial genomes, often derived either you download them from NCBI or you sequence your microbiome. And then people are looking for these biosynthetic gene clusters that produce some secondary metabolites, which are of interest. So example could be antibiotics, but there could be many other biomedically interesting compounds that we can find in these bacteria. And you can obviously validate them by other means. And there are a huge number of tools that do that. Here I have PRISM, that slide from that publication here. We use actually anti-smash from another publication, now already in version 6. There are already 6 papers out about that. And they identify these biosynthetic gene clusters and then also cluster them across species to see which are common forms and which compounds they might create. So that's highly interesting. These methods, you can divide them roughly into evolutionary, like phylogeny-driven ones, and non-phylogeny-driven methods. But in general, they all use genomes as input. And the better the genome, the better the predictions will be. And then they often have some sort of clustering into BGC families across species, for instance. But that brings me to the topic.
Stephen, just a quick reminder of the time. You have another 2-3 minutes.
Yeah, then I will finish in 2 minutes. So the problem here is that when you predict these from genomes, you first want a large number of microbial genomes, but more important, they should be complete or at least have very long contiguous scaffolds, because if your contigs break between these BGCs, they will be hard to predict or to find. So the more complete your genomes, the better. You also want unknown species to actually find novel stuff. And that's mostly these unculturable species you can only get from microbiomes. So you finally want to sequence really highly diverse microbiomes, like soil microbiomes, to find as many as possible. And the general solution to that is, again, ultra-deep sequencing, because the deeper you sequence, the better will be your assemblies, the more stuff you will find, and the more comprehensively you can describe your microbiome. So that collaboration is with Nadine Ziemert at the university. She published that study recently where they looked at a soil microbiome from Tübingen, well, multiple, and used them to identify secondary metabolites. In that study, they used Illumina combined with a low coverage nanopore. And our question now was, can we actually saturate that sample by going extremely deep with nanopore? So the overall project is metagenomic sequencing with nanopore, shotgun assembly, detection of BGCs, and then clustering of them in families. And typically people use like a 5 gigabases to 10 gigabases of nanopore. We took a single sample here in 4 replicates and by now have roughly 140 gigabases. We are still sequencing. Our goal is 300, so that we have about 50 times more than people usually sequence these microbiomes to. The quality is good. As I said, we get between, well, that one was not so great. 20 gig is not great for PromethION run, but 60 gig is already quite good. So that was a good run. We still try to optimize it to get 100 gig out per run. And we get a fairly good quality for an R9 pore. With 13, we will switch to R10 so that we improve the quality further. Again, these are 4 replicates of from the same soil sample. The read length is also not bad. We get a lot of reads beyond 10K, 10 kilobases, so that's quite good. And we have already a finished pipeline for that, where we can use assembly with Metafly, and then, as I said, anti-smash to identify known and novel BGCs. We can check the completeness of the known ones, we can check novel ones, and so on. The assemblies are still quite a mess when you look at them in the assembly graph, but when you look at them in the Quast assembly quality, you see immediately that in each of the replicates, unfortunately, we didn't compute them together yet because our computer was not strong enough. We need 1.5 terabyte of memory. We have one now, maybe in 2 weeks. But even just using the replicate separately, we get completely assembled genomes. Like here, the largest one is 4.4 megabases. And in total, just out of these best replicate with a 60 gigabases sequence, we get more than 4.5 gigabytes of assembled genomes. So that's quite a lot we see here. And it will further improve when we combine them and add more sequencing power. So to come to an end, I only asked, what is this largest contig we found, this likely complete bacteria? It's definitely not published yet. So at least there's no full chromosome published. The closest we can find is like 50% coverage, 88% identity. So it's a bacterium which has no published complete genome in NCBI. If we look at anti-smash, the 4th cluster here immediately jumped to my eye. It looks quite interesting. It has a lot of core biosynthetic genes and several additional biosynthetic genes in a nice cluster. All this stuff might, if you come from the field, will be known to you. But we can also look at this biosynthetic gene cluster if there is homology in other species, and we do find them. It's not super high, but it's there. So even just looking at this first genome, we might already have found some interesting new BGC. In total, just in this replicate I talked about, the deepest replicate, we found 12,000 of these BGCs, and now the big work starts. This data is really just 1 month old. We will have to screen through all of them. That will be quite a hassle. And there's quite a lot of bacteria we find that really just roughly...
Sorry, but your time is really running out.
I'm done. So we know that about at least 1/3 of the bacteria we have there do not have a published chromosome in NCBI. Just a last slide teaser, all of these stuff we have, all of these methods you can find in the tool ONT Tools, which is on GitHub as open source. So with that, I want to thank all the people, especially my long read and liquid biopsy team, and you for the attention, and sorry for slightly being over time. Thank you very much.
Thank you, Stephen. Time for 1 or 2 questions. I'm told that there's a question from one of the online participants. Scott, if you're there, please go ahead.
Yeah, Stephen. So I'm wondering, PromethION says 200 gigs for flow cell, and you're showing me 38 to 68. Can you comment on that?
Yes. So we already saw when in the last project that on the min ion, they also got less than the optimum from a min ion run. I guess this is probably sample specific, although I have to say that our PromethION flow cells never give us 200 GB. So for very high quality human blood samples, we get 100. And if we are very lucky, 110 or 20. But every second flow cell, we see 50. So it's not that unusual. I think either the shipping of the pores or something of the flow cells makes them worse, or ONT is always showing their best flow cells in their advertisements. Don't ask me. We get between 50 and 100. And for this soil sample, we are a bit lower than we hoped for, but I assume that the DNA extraction, there might be some stuff in there remaining that clogs the pores or something, so that makes it lower output. Yeah, I also hope that with 4 flow cells, we are already reaching the 300 GB, but we didn't.
There's one question here from the audience. That'll be the last question, and then we'll stop.
Hi, Stephen. Great talk. I'm wondering if you can comment on some of the new sequencing platforms that are coming out, particularly from PacBio, that are promising, much higher quality, and the implications that will have for your deep sequencing method.
So in general, we use both Nanopore and PacBio. In fact, for perfect human assemblies, we combine PacBio and Nanopore because of the ultra long reads from Nanopore and the much better quality from PacBio. But PacBio, as you said, always had the problem of yield. Our Sequel 2 can only sequence like 300 human genomes per year, while Nanopore can easily do 6,000. So with this new platform, they announced, I don't know exactly how much more, but it sounded like 20 times more, at least, coverage per run and much more output per year. We would definitely prefer the higher quality, especially for diagnostics. It makes a huge difference. You just get much better quality of your assemblies. You don't get more contiguous, longer scaffolds. That is more depending on Nanopore. So for the specific detection of these BGCs, I'm not sure if it helps that much, because I think a few errors don't matter for the prediction of a BGC. If you assemble a genome because you want to find rare mutations, then it's extremely important, and I would probably switch to PacBio. And as you know, Illumina also with the NovaSeq X will now again triple the throughput and like almost have 2.5 times lower cost. They will be released in NovaSeq X in January, and that again will push the ultra deep sequencing a lot, because if you can sequence a human genome for $200, you can sequence a lot of microbiomes and deeply, in my opinion, or viruses and so on. So I think it's a very good time for these ultra deep sequencing methods and to establish them in the lab, which, as I said, for me, most important are these UMIs. So that's something we are looking into, to have UMIs for Nanopore and PacBio. We haven't done that yet. Manpower is missing, but it would be great to include UMIs also for the long read sequences.
Thank you very much. Let's, in the interest of time, bring this session to a close. Thank you, Stephen, once again. Thank you all, and thanks once again to all of the speakers of the session. And we break for coffee now. Thank you.
Before going for a coffee break, I request Dr. Venkat to present a memento to Dr. Narendra. Now I request Dr. Narendra to present a memento to Dr. Karthik Raman. We'll break for a coffee and be back at 11:30 a.m. for the next session.
Okay. So we'll have our panel discussion now. So I request the speakers to come.
Oh, come on in. Karthik. Nick for space microbiome, please come to the dais. And Nikos, please come to the dais. For plant, Dave. For fungus, it's you, come. Yeah. Yes, yes. Yeah. One more chair. Take your seat there. Okay. Okay, so we are here mainly to initiate some sort of discussion. Karthik brought us here. Normally what happens, we come here and then speak and then leave, and you guys are also forgetting. I know one or two people will getting the email address and then get connected. But what I want is India is rich with talent. Okay, you guys can, unlike 40, 50 years back, we all going out and then doing things and then never return back, including me. Okay, so now that is not necessary, because everything that you dream of are going to be there. So this is a chance for you guys to ask more questions, and especially I'm going to ask younger generation to start with, like a student. Where are your students, Karthik? Can you ask Pratyay and all those things who are involved right now to come into this room, please? Pratyay, Shobhan, and Amrita. So first 5 minutes, I want the students to ask whatever questions that you have, either fungal stuff or space stuff, plant stuff, and then computational stuff. So they'll be here to answer. Is there one more chair we can put it for Coral? Yeah, sure. And since we don't have space, we can directly ask for the Coral thing also, that we have Alex on the back, and also we have Fatih, who has a rich experience with developing things to go to the space, and also he's also a project scientist for several space related work. So don't feel shy. Okay, this is the time that it will be recorded, and we have a person recording all the things, so it will go into the minutes about things, so anybody can go back and then learn what to do next. Okay, you can ask anything you want. Come on, let's hear from students. If not, I'll pick the names. Oh, here, tell your name and your affiliation, and the question loud and clear, please.
Yes, this is Sai. I'm affiliated with Dr. Karthik Raman, sir. And my question is, first question is to you, sir. So basically, yesterday, to put a long story short, you showed a machine that could process a live organism and give you as an output of processed DNA/RNA, which can be fed into this minions to give you sequencing. So basically, what takes months, it can give you in hours. So the time frame. So so is it when I asked you about this, you told me that the interface between the machine that gives you the processed DNA and which actually subsequently sequences the DNA is not there. We need to build that. So is it possible to, and it should be done aseptically, of course. So is it possible to use our HPLC automatic injection based technique to inject the DNA from the output to the input of Omicron? Sorry, Minion.
Yes. This is the kind of thing, we wanted to get into, in the sense, Fatih, come in here. Alex, if you can, please come to the desk whenever you are able to come. Okay. Okay. This is exactly the things that we wanted to hear from you, in the sense, you got an idea saying that we have 3 different modules: sample collection module, sample processing module, and data detection module. Okay. Sorry, not data, sample detection module. But these all 3 different modules can be integrated. That's the question. So the answer right now, maybe to some extent, Fatih can answer that because he was involved in connecting all these 3 together. But how you can miniaturize later, that you can take to deep sea, for example. If not into the sea, you can get into the, at least your ship, research vessel, etc. Or it can go to the doctor's office to do, collecting for COVID and other things, right? So you collect the samples, instead of waiting, you will get the answers in few hours.
I was thinking of my lab, our labs.
That's right.
Yeah.
It's not necessarily lab. You have to think about how you can take it to the next generation. Right now, 3 different modules are there. How it can be integrated so that it will be useful. That's the engineering question, also integration. Okay. So IIT is known for this kind of thing. So you young mind can come up with a very innovative idea. So how 3 modules can be integrated?
Great question, and definitely we need talented scientists and engineers to be involved in those type of projects. From a flight perspective, I think if we remove the astronauts for the equation, that would be easier and better to implement flight experiments. More importantly, that will decrease the amount of contamination, that will decrease the amount of mistakes, and also decrease the amount of resources which are needed. So currently we are having problems of the fact that we are in roughly a 5-year cycle. So it takes 5 years for someone to be selected to test and validate an experiment, to implement the experiment, to conduct the operation, to wait for the samples to return, and then perform the analysis. It's 5-year cycles. And so I always say to students, if you are a PhD student, there is no way that you should be working on one flight project only, because the likelihood for that project to be delayed, cancelled, and so forth is very high. And therefore, you need to have more, type of projects that you are undertaking. And so that being said, miniaturization is very much appealing, and automation as well. Miniaturization from a resource perspective, because as you imagine, we're kind of transitioning from the International Space Station to go beyond low Earth orbit, and so going back to the moon and beyond. As you can imagine, volume and mass will be an issue. Similarly, from a microgravity perspective, if you have instrumentation which are microfluidics based, that also help very much the processes, because then you reduce the amount of volume, but certainly you reduce the amount of impact of gravity on those processes. I didn't show you, but actually I do have a video of Kate Rubins performing and using the mini ions. And actually you will see her, she's taking the time to swing the samples. And the only reason she's doing that is she's just trying to remove the bubbles which are in the samples. And as you can imagine, bubbles in space is a big problem, because you don't have convections processes and everything is kind of based on surface tension. And so if you remove that process and you have instrumentation which is microfluidic based, obviously you don't have a consent type of issues. And so definitely those type of technologies are needed so that we're not heading ourselves into that 5-year cycles, but then we can more efficiently, one, perform investigation in space, do the analysis in flight, get results immediately, and then you can refine your hypothesis. And so instead of having that 5-year cycle, now you're kind of more on something where you have much more quick turnaround, and which is kind of very much similar to the type of investigation you do on the ground. Sort of. Right. Right. I mean, sure. Yeah. I mean, having end-to-end processes is the key there. And so
it's been automated fully.
those are type of technologies which are needed. So I give an example, which is GEM, stands for Gene Expression Measurement Module. So again, we didn't test it in space, only did testing and validation in the lab, but that system was end-to-end. So we grow cells and then everything goes from lysis cells, extract nucleic acids, hybridize it to an array, and then you get the results. So those are the type of technologies which are needed, and certainly integration from all those different steps are critical. And smaller automations and miniaturization are key.
Yeah. So from my understanding, you were asking if you need to separate DNA from RNA actually to do the sequencing, right?
No. Actually, my question was to you have the cell/cell lysate. So I inject it, and instead of outsourcing it to some company which is going to give me the data in maybe 2 weeks or a month, I can get it by the end of the day. So my throughput will be that much higher. So that's where I'm coming from. And if that to as a follow-up to your point, so if that machine has already been integrated, what would be the cost of the machine if you mass produce?
Yeah, I mean, so and again, this is a problem within the agencies, and I think space stuff is important, but I think you kind of need to look at bigger pictures. I mean, having something which is small that can go in the field is not just applicable to space applications. It's applicable for you going to area in the world where you have limited capabilities and so forth. So I think I think whatever you're planning to develop as a system which is automated and miniaturized have tremendous application on the field, certainly. Then in regards to your question, certainly there's a need for those type of technologies. And currently we had a design for nanosatellite technology, which is currently at TLR 5. So TLR stands for technical readiness level, and flight is TLR 9. And so we're kind of between the two. And so this is again developed for nanotechnology. So that was for nanosatellite. And so that is why there was some constraint for size and dimension in particular. So it was 10 by 10 by 10. We do have a model for the International Space Station, and roughly the cost per sample to be processed was roughly 4 or 5 per sample.
And the cost of the machine, sir?
So the machine is kind of built in a way, and so that was the total cost for the device per se. And then you just need to add an instrument to kind of be able to monitor the environment, so temperatures and things like that. So that is available on the station. So the idea was to have a system which is kind of have plug and play. And so roughly our estimate was, a few hundreds of dollars per sample.
Once again, I think what his question is, right now you cannot go and buy. Okay.
Yes.
That's one thing. The second thing is, you engineering mind can come up first to understand the basic question here. Sample collection sample processing, sample detection. So these three has to be integrated, which is, and also validated, like what Narendra Dikshit showed, that the computational model, but he did all those things as, mixing, matching different kind of microbes. So the end product that he showed what is predicted, what is there. Likewise, you have to come up with an engineering idea or collaborate with the engineer. This is what is a very good discussion going on here. Collaborate with the engineers to make your dream comes true. Okay, it is possible. Is it there? Majority of us thinking it is not, but we may have a single prototype that what he's talking about, and that I want Nick also to add one or two comments on this.
Yeah, I would also say, it's from and Fatih was talking a little bit about this as far as the applicability, right? And this is kind of one of the areas for you to really think big when you're thinking on this stuff, in the sense that, we've heard everything from, E. coli cells to DNA should be running nonstop through this, to wipe and swabs, but we definitely have needs for everything from soil regolith to built environment to swabs to air. So multiple formats should be going into this, not only for space application, right, but a lot of this stuff you can use on Earth. And so I think it's a really broader solution here for even potential everyday life that impacts us.
The answer to that question?
Yeah. Do we have an integrated system to monitor microbe plant?
We do not. Yeah, we do not. Everything.
You are a private company. Are you in the process? Are you the people are in that area? Are they developing something?
Not really not from a microbiological perspective. I mean, there are in plants, we tend to monitor plant health, and the proxy for human eyes is like either multispectral or hyperspectral imaging, where you can image the plants and determine the health of the plant, and even apply analytical techniques, machine learning to that, to automate the analysis of the output. So that's something that exists, again, large scale. I mean, they have these on planes and on satellites that fly over and look at crops, but we don't have something that's this big. It's desktop.
Let's hear from DOE. What not necessarily, you are a computational biologist. I understand that you have, once the data comes in and you know what to do, and however remote it is, you know what to do. But do not only your group, is your agency is coming, for example, DARPA and other people that you know of, to answer this question?
Yeah, I'm not sure. I mean, I'm not sure how much it applies in our area or in what we do. So I mean,
go to the soil, so bringing it to the lab and then get extract the DNA.
Yeah.
If you extract DNA and then do it.
That will definitely be helpful. Yes. So, but
anybody working?
not that I know.
Yeah.
Yeah.
Coral, let's say here from Saudi Arabia.
Thank you. Yeah, I just want to agree with this, the need of interactions, really strong interactions between the basic sciences and the engineering, because when I think about try to scale up, such process or measurements or and something like that, there's no way. I mean, I can relate with our work with probiotics in corals or mangroves. So we are talking about a process that we apply in a small scale, and it's working perfect. We are going now to the field, and so far it's working, but it's still a small field. When you think about, scale up this for huge ecosystems, and so it's another story.
So there are other questions will be also there. Here I wish to ask eminent, institution like IIT to come forward to see whether you have an answer for this question. Go ahead, Vijay.
Hello. So myself is Kedam from IIT Gandhinagar. So I work on molecular biology, structural biology, and so on. And also is part of the central instrumentation facility there. And to this particular question, where the integration of all sequencing and as well as isolation sequencing and immediate results, I think India has a vast instrumentation facility, and integrations can be done very quickly, and it is all available for everyone in the national portal, where you can use isolation facility from different institutes. You can also do the sequencing in the same institute if it is IGIB or CCMB, where most of the biological work goes on. So we can definitely have this integration done there itself, then and there itself, where you can stay there, do the experiments, isolate it, sequence it, because it is open to everyone. It is national wide. It is called ISTEM portal, where if you can go there, you will find almost all the instruments, let it be a material science instrument or a biological instrument.
The question is,
yeah,
where can you integrate and then field deploy? That's the question.
It can be. It can be.
That is the thing needed.
Yeah,
Okay?
it can be. It can be done. Only thing is the approach that is taken towards connecting people at the right institutes and at the right place is needed.
So that's a very good, and get connected.
That is a great opportunity to do that.
Yeah. Get connected with him. So two IITs can do something together.
And one more thing is anyone can register for ISTEM portal. And also you can talk with the technical team over there to connect with the institute run central facilities and get it done. Or you can go there and do it. That is how.
Okay, since I gave a very short answer, let me add a couple of additional things. So one is, I totally agree this is extremely important to be done and it has not been done, but also in the process of doing that, standards have to be developed, because there are no standards in isolating DNA or even in sampling. This is one. There are international consortia or communities that are trying to agree on some standards, but there are no standards yet, even on human microbiome or in general in environmental isolation of environmental samples. And the second is, we don't even have one of the most important things when you're doing the sampling and extracting DNA then, etc., is to collect metadata at the site of the extraction of the sample. So these are not going directly into any specific database right on site. It's written probably on paper. And then when we are asking the metadata to be submitted with the sample, keep in mind that the sample is completely useless unless you have metadata, right? So if you give me a sample and I have no idea where was the location, etc., what were exactly the exact parameters and so forth, it's for comparative analysis, it's very close to useless. And that's why a lot of data sets as well in SRA that they don't have any metadata are completely useless. I just see sequences that I have no idea where they came from or what were the parameters. So even that doesn't exist right now.
That's absolutely right. Otherwise it become garbage in, garbage out. So you think that the instrument is giving you everything. Okay. So it has to be carefully planned. But first thing is you need an instrument, integrated instrument that is not existing, that you can take it and use it today. Okay. So let's move on to other thing. Because we already spent around 25 minutes laboring this particular topic. Microbial monitoring, instrumentation, integrated instrumentation is needed. That is the kind of verdict based on this discussion, unless the audience have any kind of different opinion. Okay, it's in silence or no. Let's talk about the other one is, biosensor development. You have heard so much gene-based analysis, okay? Gene-based analysis takes around, right now, even with the integrated system, it takes around, 1 human work day, okay? 8 to 9 hours. Can we sense microbe? That's another big thing that young mind can think about it, because specific bacteria release some volatiles. So instead of taking and extracting DNA, can you come up with some sort of, small biosensor? So we call them electronic nose. Can you use that? Because those electronic, you have a smoke detector even here in your home, right? That likewise, if you have sensing nose, which is electronic nose, for certain kind of, you cannot detect all bacteria and fungi because bacteria and fungi are everywhere. Okay, but at the same time, you have E. coli, for example. You have, it's called escape pathogen. That means the World Health Organization is already declared what is escape pathogen is. So you can come up with something related to that. I think Shripriya is working on that. Do you want to say something about where we need to go from where we are?
Yeah, good morning, everyone. I'm Shivapriya. I'm a faculty at IIT Gandhinagar. So I'm basically an organic chemist. Okay, so it's absolutely possible because the molecules that secretes or that forms the biofilms or whichever is needed for the bacteria, if they can be characterized, that's very important first. Okay, so there are small organic molecule dyes, which is used for several applications, even for cancer detection. So it should be possible if we detect the biomarker of that particular bacteria carefully and characterize them. So it should be possible. Yeah, but we need to work on it. But can I also have a question?
Yes, go ahead.
Okay, so I have come here to study how will be the future of medicine, okay, in space. Okay, so that's a main goal of me coming here because I work on therapeutics, especially for the pathogens which causes cancer like HPV or H. pylori, which causes stomach cancer eventually. So I work on those pathogens at the molecular level and try to develop therapeutics for particular protein or enzyme, which is very important for the survival of those pathogens. So here is a question to the plant biologist or the plant person here. So A. thaliana has a gene called TLK, which is very important for its flowering. The same TLK is called tosyl-like kinase in human, which by overexpression causes glioblastoma or ovarian cancer. So that's proven very well. So it's just 15 years old kinase, which is discovered 15 years back in human. So is it futuristically, are we thinking that if we have these plants grown under certain conditions, will that be effective or will it have any adverse effect on human? Eventually, if this particular gene overexpressed in this plants, will that become toxin?
Very good question.
So I'm again going to preface this by saying plant box engineer, not so much plant biologist, although I did not stay at a Holiday Inn Express, so I don't really know the joke. But anyway, so it's a good question. I really don't know the answer to it, but I would say that it's definitely an intriguing thought that would need to be sensitive. If I understand your question correctly, the question is that perhaps over long-term exposure to Arabidopsis thaliana, you might induce cancers in humans. I mean, in a closed environment of like the space station. Is that your question?
or by consuming. I mean, not only for A. thaliana, for other plants that we are planning to grow in space, and if there are mutations which would probably induce these kind of genes into them, Okay, and then by consuming them, will we have any effect?
I can certainly see that as being a threat. I mean, there is always a concern when we take any biology into space that there's going to be that the biology is going to respond to this novel environment of spaceflight in an unanticipated way. In fact, that's, at least for plants, that's a lot of the reason why the plant researchers want to take plants there in the first place, is because, as I said in my talk, no plant in the entire, history of plants has ever had to cope with the microgravity environment of space. They've all grown, terrestrially here, and so it's not always known what will happen when you take them to space. But actually, you brought up an interesting other thought that I had, which was, there's a lot of discussion. Actually, this is playing off of what you had said about an electronic nose. There's a lot of discussion about using plants as a canary, as a way to sense, as a biosensor, sense the spaceflight environment. Again, it's usually coupled with something like multispectral imaging of the plants, but plants can be either on their own or engineered in such a way that they can, say, fluoresce when they detect a certain contaminant in the environment. And to Fadi's point about resources, for us as engineers who design stuff for space, it's always about resources. And so if the resources involve, so let's say, for example, not Aethaliana, because you're not going to consume, tastes terrible. I don't know if you've ever eaten it, but I did on a bet once. But anyway, if it was lettuce, so if it was something that you were already growing as a staple crop anyway, but you could also use that lettuce as a biomonitor of what's happening in the closed environment of your spacecraft, well, then that's there's really no additional resource. The only additional resource is simply to drive the analytical tool that is sensing the plants, for example, as opposed to having an entirely different, say, electromechanical system, which itself can fail over time, may have different sensitivities. I mean, you talk about one last bit about the electronic nose that really made me think is, people use dogs these days to sniff out cancer in humans. The dog walks through a hospital and can sniff, and my understanding is because the dog is so very sensitive to, these molecules that are given off by the cancer cells. So all this is very intriguing and perhaps something that young minds can capitalize on.
Yeah, if I may, actually, I will reverse the question. Certainly, your question is valid and intriguing because certainly you have concerns for the health of the astronauts, which is obviously important. I will challenge you to reverse the question, and instead of specifically only addressing the health of the crew, what are the current capabilities of using microbes or plants and other biological systems that drive this application for you? So, for example, to try to transition to my world. Which in particular which towards the use of microgravity, space radiation, secondary metabolites, and to find some interesting compounds that might have some biotechnology or human health or pharmaceutical related applications. And this is much more important in regards to application. And I think us transitioning from the International Space Station to those commercial platforms, part of the work would be, scientific related for NASA, but the bulk of the work will be specifically devoted for commercial application in pharma and biotech. And the idea there is to use the uniqueness of a space environment to try to come up with interesting answers to address antibiotics resistance, to address cancers, to address immunocompromised individuals, and so forth. And I think this is one of the from a space perspective, this is one of the most important aspects, I would say so.
Thank you for the, so basically, drug discovery will be helpful. So you have a, this is to promote science, the microbiome science in India. Okay. So keep asking questions. So what are the things that India is lacking in terms of some academic, teaching certain aspects. So one of the things I think a lot of people are interested here is, have the awareness of the astrobiology in India. Okay. So let us hear from Bala about that. And, please ask the question short because we have to cover a lot of things, and the answers also will be short, so that we can cover more people. And please start thinking about, several aspects. Okay, not only, science, it can be something, communications and other things. Go ahead.
Yeah, okay. I thought of, in fact, asking you, what is the way forward for astrobiology in India?
In India, okay.
Because we've been trying for several years now to bring
Yeah.
a group and so on.
Yeah. So the, premier institutions and even, other academic, institution in India should also think about that. In other words, including at least, 1 or 2 credit kind of thing. So not, a lot of teachers are here, academicians are here. They can tell to their dean and others, that's how it started with the Indian Institute of Science. Now we are teaching it. And everywhere I go, they are also interested to include in their curriculum. So it is in actually your hand. In other words, we back in US also not having that as a main curriculum, because everybody nowadays, the academic institution is business tailored. I go to this course, when I get out, can I get 1 lakh or 2 lakh per month job or something? So astrobiology is a lot of unknowns. Okay, so it is more research oriented. So instead of teaching, what you can start doing it as a part of their PhD program. For example, now computational science, Karthik brought this, space in it. It become more sexier because he's validating his model into the samples from space. So that kind of collaboration is always possible. That brings awareness to the students, and then they can learn things. So that is the only way. Even if you go to universities in the US, you don't have an astrobiology, masters in astrobiology kind of thing. It is a course or a kind of internship going on. Okay. Let's hear from something from a medical institution. Okay, Damu is here.
Venkat, I just had a question, right? So because we have people from so many different countries on the dais. So one question is, given the current state of microbiome research in India, what would be the best way forward to sort of ramp it up, right? From experiences in your own countries. Like, for example, I see the National Microbiome Initiative and things like that. And maybe Alex can share what they've done in Saudi Arabia. So what are the, key things that should be done from a policy perspective or from, top down, right? So like, let's say we had to propose something to the Department of Biotechnology. What should be highest on their priorities?
So a couple things with that. I think one of the other keys, and this is something we're very much looking at to start establishing within the US and particularly for NASA to help solve these problems, is really look at kind of our communities of practice and having ways to, network people through these kind of complex disciplines, whether it's through series of workshops at various strategic points, a symposium website, kind of Slack channels, all sorts of venues to be able to, ensure that we're getting our stakeholders and our end users and researchers kind of the latest publications. For example, we just had a workshop last week where we looked at kind of the research side to the engineering applied application. So really trying to have a means to kind of put these communities together that wouldn't normally be in a session like this or go to the same conference. So trying to find those opportunities, I think, is really key.
Thank you. So first, I'd like to apologize because I was late for this table, because my son was defending her master thesis, and I was trying to watch a little bit. And I will take a ride on this. My son defended the thesis today, and actually change a little bit the debate here. It's about the human resources. How about human resources in India to work on microbiome science? Because microbiome science now, as you can see here in this table, and also in the audience, is not, it's totally inter and multidisciplinary field nowadays. And we need people with different training. And in Saudi Arabia, we don't have this, and I'm trying to push this microbiome field in Saudi Arabia, trying to train and to get more different expertise together, so like people from the computational science, people from the microbiology and the pure microbiology that we always stress this point that is so important, and the omics, all the different fields. And now we can go to environment, we can go to gut microbiome, we can go to pharmacomicrobiomics, we can go to drug discovery. So it's a huge field nowadays. And
yeah,
that's an important aspect, human resource.
That's correct. So, if to add something about what the question is, so what they can do to move forward this in India. I don't want you to repeat exactly the same thing what we have done from other countries. So you can learn that and adapt, but what is needed has to be identified. Right now, what is needed is more engineering aspect. In other words, how you can get them to answer. So I don't want you to see, something like, go and collect samples from various different places. More than that, you do that with appropriate instrumentation, with more mobile lab. Okay, so she's having some question.
I wanted to comment about the astrobiology aspect in India. So I am a member of Astronomical Society of India, and it is a very well established and 50 years already and powerful organization. So why, and we tried for a few years to establish Astrobiological Society, but it seems to me that astrobiology as a subject is very reluctantly accepted in India.
Because there's no job market.
Well,
I'm not joking. That's one of the reasons, it is tough, but it is very highly attractive. And immediately the funding people are asking, okay, you teach this, what is next? What is this person is going to do? Right?
Yes, and
so we don't have any kind of flights to go and then look for life in other planets. So, so you
we
have
still
that.
do, but again, it's again done from astronomical point of view. In the side, we have exoplanet group, we have small astrobiology, small group. But when you go to DST, let's say, talk to DST, astrobiology, they will laugh at it.
That's right.
Really.
Because you have astronomy, you have all the images come to you. So you know how to look at it with the computational modeling and mathematical modeling. So you will be looking at those things with the finer details. But when it comes to the astrobiology, that is different, like what he said. Do we have life in the earth? The Galileo is not saying exactly the answer. Maybe, right? So that is the problem. There is a problem. Now, as Fatih also said, by doing astrobiology, space biology kind of thing, if you find a wonder drug, then immediately it clicks. If it comes from India, for example, that's where all the young students have to think about it. Okay, so you don't need to delve into and do for your life that, but if you are able to find that, and then you will increase awareness. What you cannot do on the earth, if it is done in the space, that's different.
So following up on Karthik's point, actually, I wanted to ask the same question. What should be done in India, or what will be the future of microbiome research in India? And thinking about it, obviously, India has enormous resources, human resources, and very strong individual, very talented individual groups. The question is, are there community efforts? Are there any national initiatives to bring those outstanding, let's say, groups together to address much bigger questions? And this is the tendency across, in the entire world. We're moving away from individual PIs. Individual PI research obviously will always be there. But in order to address bigger questions, we need communities and we need bigger initiatives. So following up on pretty much on Karthik's question, what will be important? How can India be a major leader, protagonist in the microbiome research globally? Or what is also important for the national economy? Are there specific, can you identify specific focus areas or challenges that will have a tremendous impact on the Indian economy or Indian society, and organize big initiatives, national initiatives around those? Actually, we have done something like that in Greece. The initiative is called Microbiocosmos, 3 Greek words, micro, bio, cosmos. And what is one of the most important areas in Greece is oil. Greece is a very important oil producer. So, and the Mediterranean diet is also very well known. So there are compounds there that they are very important for longevity and other parts. So there is also a very important issue of contamination of the environment in the process of oil production. So these are areas then that the microbiome community, let's say, has come together in order to come to get funding, national and European funding, in order to address these big initiatives. So my recommendation or my suggestion would be to identify big challenges or big questions, grand challenge type of questions that will bring together the community across all India to address that, rather than going after individual PIs or individual groups.
I'll stand up here. Wow. I've just been listening to this and have kind of 2 thoughts. One, you kind of want to think about the incredible genetic diversity and microbiome diversity that's present in India. And I think if more people knew exactly how much diversity there is here, they would want access to those samples. So it should be like in your mindset that this is a national resource that you want to protect from people coming in. We're going to survey Indian microbiome. You're like, no, that's ours. It's an incredible resource that should give amazing benefit to Indian health and science, and that you don't want other people doing that research. So it's another motivating reason to make sure that everybody's trained to be able to handle that. The other thing is, I think listening to everybody over the 3 days here, the one thing that we see in microbiome research is that it's very, very heavily molecular. Venkat's probably one individual who's made an incredible career out of isolating microorganisms and doing experiments with those organisms that have biological relevance in astrobiology, for example. And that it seems to me that cultivation microbiology is a niche that could be exploited because it's being neglected elsewhere. We see that, from my own career, it's the easy, lazy way to get data. You're like, okay, I've got samples, I'll extract DNA, I'll run a PCR, do sequencing. I'm going to have data in a week. It's intoxicating, but it leaves you with an incomplete picture. You know, I was just saying, you detect a shift in a microbiome based on DNA, and you say, I think this organism does this, and that's why we see this. But there's enormous gap between the DNA results and the physiology. Are the genes that you're thinking, are they expressed in situ? Are the metabolites there? Are they interacting with the host? There's an incredible range that you need cultivation-based microbiology. And maybe that could be a niche that could be exploited here.
Good. I think, you know, once again, I need to give some thing for everybody here. So get prepared, and I'm going to call one student, think about what you wanted to do next, because we have only 5 more minutes to end up this session. So he also have one question. He's asking me to give the mic. After that, Fatih, please ask the short question.
No, it's not really a question, but a comment, because I see that India and Brazil have very similar situation right now about, we have incredible diversity. We have very talented researchers. We have good initiatives, but I don't think, another thing that is really important, it's convincing governments that funding for those areas it's very important too. Because we have very same problems, I think, and very good, we share very good situations too about, you know, diversity and researches, but actually funding, it's very important too.
Thank you. Short question, because we have to move to some other students.
Sure. Maybe some final thoughts, if I may. And so, certainly, for where I do see 2 approaches, one which is top down, and the other one is bottom to up. I think as a community, and there's talents, and all of you are rock stars. And so I hope that every time you wake up and you look at yourself in the mirror, you recognize that you guys are rock stars. So that's point number 1. I think us as a community, and I'm hopeful that we will get together and try to lay out some foundation in order to publish a report or publish a paper that kind of summarize all the different type of science and interactions that happen during that 3-day symposium. And that could kind of lay out some foundations, and then we can reference that particular paper or report to be able to seek more funding for you so that you can perform more science. For example, 10 years ago, we had that instruments, which was sponsored by astrobiology program. So we received $1 million to develop a particular prototype to, again, do end-to-end processes to sample going from culture to analysis. And then that device is life science, and therefore the transition from astrobiology to space biology and human health is pretty straightforward. And so we submitted that to NASA, and actually we never received any feedback, never was reviewed. And then very quickly I understood that actually they didn't understand what we were trying to do. And then I took the responsibility to educate NASA, to educate manager, to educate people that make decisions. And so I wrote with collaborators of mine a review describing what is omics, describing the power of omics and how those type of methods have tremendous application and tremendous power for human spaceflight and space in general. And the whole idea there was to establish a foundation so that those managers, which are decision makers, understand what we're trying to achieve. And so I think also we do have a role to help decision maker to understand the importance of the type of work that we are currently conducting. And then there's the other thing that Stefan alluded into, which is pride. I mean, Indians are very proud, and you should be, because you guys are a wonderful civilization. And as I say, all of you are rock stars. And I think that if the top-bottom doesn't work, then it should be bottom-up. And what I'm trying to convey there, then show them that things are possible by interacting with others, by collaborating with others. And then maybe you would be focusing on a particular niche that suddenly blow up. Like, for example, Personiae, which, was an organism isolated from the International Space Station. And suddenly you're realizing that particular organism now has a tremendous impact and has been collected from patients in different places in the world. This definitely blow up, even blow up myself. So then if you work on a particular project that suddenly has like tremendous media or tremendous impact within the community, then politicians will start to pay attention and say, hey, see, there's things that we can do. And therefore there will be that Indian pride that will be coming in, and therefore more money will be pouring in. And so I think there's those two approaches that have to be considered moving forward.
Yeah, thank you, Fatih. And along the line, I want some students and others to, to ask question. Why don't you give it to her? She's asking. Yeah, go ahead.
Good afternoon, sir. Myself Shraddha.
Mic closer. No, closer. Yeah.
I'm Shraddha Sharma. I'm PhD first year student. So
Closer to, yeah.
my question to Dr. Marcus, actually. Yeah, so my interest basically is in bioremediation, and whatever literature that I have read is mostly focused on using prokaryotes for bioremediation purposes. But I rarely see fungi being used for that purpose. You have done a lot of studies from Brazil, and you know that like you have isolated a lot of samples. So my question is that have you encountered any species that has this good bioremediation could be used for the purpose, and during your research that has this?
Yeah, so if I understand, if there are species that can be used for bioradiation.
Bioremediation, yes, sir.
Remediation, yeah. Yeah, so, that, so fungi are very diverse, right? And there are many species, and especially with now that we have more genomics capability to understand metabolism of those fungi based on genome research. It's a growing area, right? And I think that many species of fungi, specifically Ascomycete fungi and some Basidiomycete yeasts, they are very, very useful, not for only bioremediation, but for many other different applications. And yeah, and that's pretty much the answer, but yes.
Once again, thank you very much. And let us ask a question about what you guys can do next on the microbiome research. Go ahead.
So my question is related to like the uncultured fraction of microbiota. That is, there is a lot.
Once again, what India can path forward? Directly one specific question that you have, you can catch hold of them and then ask what India has to do. What are the needs in this microbiome research?
Okay, so yeah, I would like to point out that we can take lessons from the genomics community. Like whenever I'm using a tool, if I have some doubt, I can ask it on a Slack channel or on GitHub and I instantly get an answer. So I think a community should be established for India, and how can we do that? So that like if someone has a doubt related to omics or like these new tools, because I see a lot of faculties are reluctant to like take up projects related to omics because they are saying, okay, yeah, that's just a computer, anyone can do that. But that's not the case, right? So how do you suggest we convince our faculties as well as like establish a community?
I got it. Nikos, you are the right person to say this. Why, what should they do?
There can be several different ways. Establishing a society, right? A new scientific society is one way, like we heard before, astrobiology society in India. It can be an omics society that will bring together people and organize conferences as well, once a year or once every 2 years, but also create a community then to have the necessary mass to provide answers as well for students and young scientists. That's one way I can think of.
Are you satisfied now? So what is the name of the girl?
I can add maybe something that for you to consider, which is GeneLab. And so GeneLab is a NASA-derived database that lists all the omics information from spaceflight or ground-based investigation sponsored by NASA. And within GLab, there are analysis working groups. So there's one in microbiology, one in multiomics, one in plant, one in animals, and a new one which is in AI and artificial intelligence. Those working groups are actually voluntary based. And so people get together on a monthly basis to interact with each other, and then they decide to kind of work on a particular topic. And so then you have people from all around the world interacting with each other and working. And so you would have experimentalists, you would have computational biologists. Certainly it's heavy on computational biology. Nevertheless, we still need other people to be able to interpret the data. I mean, certainly computational biology is one thing, but then you need to put the story together. And so again, this is voluntary based, and certainly please go to the GeneLab website and, provide your name and information there, and therefore then you can participate. And again, the idea there is you have a large network, which is at an international level. Everybody has different type of expertise, and therefore then you can interact with them, you can ask questions and so forth. And what is also the exciting part is that we are very, very productive, and we publish on a regular basis. And then there's also a lot of opportunities for you to kind of learn new skills. And so I will highly recommend you to consider and looking at those analysis working groups.
Thank you, Fatih. And Anurup is already a member of the GeneLab. And before giving to Saujanya, Bala, take a note of this. GeneLab is the one that in your workshop, you recruit the people who will work on those data already generated. So there's one way to generate the interest for the people. And already some of the Indians that in the students are actually in there. Anurup can give you how to get into those things also. I will come to you in a second. Saujanya first, and then after you.
Thank you, sir.
Ask the question that is what India has to do in this research. Okay, not a specific your problem. Your specific problem you will answer separately.
On an India, if I want to talk about like how India is lagging behind, I think there isn't much research that is conducted in like a microbiome study that is conducted in India, because like when I do my project, I think I'm always based on studying on a worldwide basis, because on an Indian basis, it's not really that much. So I think like from the ideas that you all suggested and from the people suggested, I think we need more of a community, like in which people from, I only got to know about these kind of works that's happening only because I came here. I think I need to know a lot more works that is done so I can explore in this field.
That's great.
I'll
Indian
just paraphrase that question a little bit. So do we need a critical mass of India specific data sets and so on, right? To really sort of, that will give impetus to because the gut microbiome project is obviously on. So you saw Yogesh's presentation yesterday, but for example, more India specific data sets, because I think, for example, one of my students has been using Bangladeshi data sets and Malawi data sets for, related to infant microbiomes and so on. So we need a critical mass of Indian data sets, which will probably push this forward. Maybe that's
what I heard is the awareness for the students has to come from the elite professors. That's what I'm thinking. You sensed and you have requested your students to work on this kind of different data sets. Likewise, the academic institution professors has to come forward, and then so that the next generation scientist will pick up and go. Let's hear from her.
Yeah, so hi, Tulika here. What I just wanted to mention as to what we were discussing in this same context, that we need a central repository for the Indian data sets. That is one essential thing that I feel, because I see the data scattered around all over and we don't have a central place. I know there is a wealth of information that we have with respect to the Indian context. That is one thing. And second thing that is hurting us probably is the open access policy. We haven't discussed about it, but the recent open access policies, particularly in Indian context, I don't know, Karthik would actually agree with me, the huge publication cost for the papers, and so that is our government is still probably not sensitized enough to fund us. So that is another problem that I see, and I think these are required to be addressed in order to build the community together in India. And we definitely need a consortia within India with respect to the human or maybe a microbiome focus.
That's right. But I take this note in the sense, if your workshop have some funding for specifically astrobiology related, I'm picking that one, astrobiology, things come out and you can say, okay, I'm funding the paid charges. So that is one way to help the people to come forward to write. I know it's $2,000 to $4,000 nowadays for publishing in a good journal. So that kind of funding, if you are, central laboratories like you have, started, that kind of pooling, it will be good. So who is next? Yeah, go ahead. Raise the hand who wants among the students, then she will identify it goes there. Yeah.
Okay, so I'm Vijay Raghavan. I work as a scientist at the National Institute of Ocean Technology. Speaking about how India could progress in this field of microbiome research, we've been talking about the consortia development or a centralized agency. I just beg to differ with the opinion which was told. I feel the data is scattered and there isn't a dearth of data, at least in the recent past, wherein the sensitization regarding the microbiome research has been receiving impetus both from the government as well as from the private sectors. All that I feel which would require is a coordinated effort from various aspects like the microbiologists, the molecular biologists, the computational biologists, the people working on the culturomics to come together and put up as a single portal consisting of something similar to the Earth Microbiome Project, like the microbiomes from the various ecosystems that exist in India, so that the students can actually get to know a picture of what is being happening in India. Not being speculative, but being on the data-centric analytics, what they could infer out of it. They can actually get a fair idea of what is being done, and they can develop an idea of what needs to be done in the future.
That's exactly what Srivastava also telling, Dr. Srivastava was telling. Exactly. You are seconding what she's saying.
Yes.
Right? Not disagreeing, right?
I'm not disagreeing.
Okay,
I'm
good.
just.
Let's move on to the other student. And next, who is next? And okay.
Hello,
Next
this is
is,
Priyankar from Thapar Institute. My question is more towards the experimental aspect of gut microbiome. In India, or any other place in the world, many people are working on this omics strategies using genomics and proteomics using already available data set. But at least in India, we are lacking in experimental, the mechanistic studies related to microbiome. And one of the, the drawbacks is in India, we don't have any germ-free facility. And personally,
what facility?
Germ-free autobiotic facility. So, we are using antibiotics to treat the mice, and which is not a good way of, making.
Did you introduce yourself? Are you a faculty?
I'm a faculty from Thapar Institute.
Then
But yeah.
go, come on, develop something germ-free facility and then write a proposal and see how is it coming.
Yeah, I mean, for that kind of facility, you need huge amount of money. It's
That's
not,
right. Think big.
Yeah.
Think big.
Yeah, for that, yeah, it's a good idea for that. You need a big group and also, similar-minded people.
So send your request to this forum. Karthik will be putting together in minutes of what is happening. So that will be, published somewhere. If not, I will publish in the Indian Institute of Science where I have the journal right now. So we will publish the minutes right there. So all of
very essential. Yeah.
all of your thoughts and others can be published there right there.
Thank you.
Okay. Then who wants to talk? That's right. After I'm ready for the next one. Anybody? Yeah. Okay. Gotcha. After
Am I audible?
Yeah. Yeah.
My name is Balu Ranganathan. I'm from a startup from IIT Madras Research Park. We work on breast cancer therapeutics. This is basically to do with microgravity. And then people have been talking about the therapeutics there. What will be the cost of the microgravity test pad in India? It's to do with astrobiology.
Yeah. It's not.
Can I just add up to one more thing?
Okay.
What we have got the information from DOD, congressionally directed medical research programs to do with DARPA. So they are now coming up with the breast cancer program. We completely work on breast cancer therapeutics to do with the microgravity environment, the test pad in India. So I am aware of one thing in Poland. I'm not sure in India. Thank you.
Correct. So microgravity, simulated microgravity instrument is also, is possible. And I showed the parabolic flight and other things, which is expensive. And going to space station through India is another, astronomical money. but you have to prove with the simulated microgravity setup. So send us information, and then I will send you back, how much it cost. And also you can do the collaborative work, and we can fix and send it back to you. Then you do the research to start with, and then show something you are. Once you are there, because you are in a institution that you are in charge, so we already have a MOU with Karthik, so we can get such kind of sample for you to start and then get initial data. Then you take it to the Indian government and see how they are funding. This person is next, right? Here is the first that guy. He was asking next to you.
Well, thank you for this opportunity. But I am
Speak loud.
Shivatsan from Dr. Ganesha's lab. So I have personally faced like kind of a trouble over computationally. Like we haven't even though IITs, prominent institutes in India have computation facility, but we still suffer a lot over computational power in terms of RAMs or cores or anything else. So I guess we need more and more computational power to access it.
Sure. See, there is this national supercomputing mission. As part of it, there is a lot of democratization of computing resources. We can talk about it. There are definitely initiatives through which this can be accessed. And
That's right.
yeah, and we are very happy to share our resources. For example, Anurup is already, using some of our resources to do some analyses and so on. So some of these can be on a one-to-one basis, but I think there are also formal channels for doing this.
That's correct. And also, Nikos already told that there is some workshop coming, and JGA is having open access also for some, you can ask that computer power, and then you will be able to do it. And here.
Yeah. Hi,
Who
I'm
is
in
next. Go ahead.
I'm Indumati, PhD student working with Dr. Karthik Raman and Dr. Himanshu Shinda. As far as I accept the fact that we need Indian specific data set, I feel like we also need a project database where we know what people are working on regarding the microbiome, because I have seen multiple other countries which has a list of projects they are working on and who to contact and all. But in here, it's very difficult to know who are the people working on the microbiome and how to contact them. That's the major problem in here.
That's right. Ask this question and suggestion, so we will put it in that minutes paper.
Yeah, just to tell you, so in Brazil we have a platform that calls Plataforma Lattes e a Plataforma, and the platform Carlos Chagas. So, and exactly, so you can access what are the main topics of research for anywhere in Brazil. So those kinds of platform definitely will help.
Yeah, he can be sharing some of the thing like for my college and other things also. They have that kind of portal that helps you to get information. And as Nikos suggested, NCBI is very strict now. Without metadata, they don't put anything here. Go ahead.
Yeah,
Who's next?
I'm Manoranjan Singh Rathod from Shastra University, working on the plant microbiome. So my question is basically from Earth to going to the space, how we can establish the plant basically on vegetative purpose, because without vegetative, we cannot survive anywhere. So the basic question is how we can develop the proper environment to this space, so we can develop a good quality and resources for the vegetation, for the other space guys.
So the future really in developing space plant agriculture is going to be on these commercial platforms. And so if I understand your question correctly, I honestly think that that the country of India could, the commercial platforms are not going to be restricted to just US customers. They will be open to, it's the vision is that the commercial platforms will be like a research park in space. So if as an entity, I think that India has such a strong agricultural base as an important part of your economy, as an important part of the people of India, that I would say it would be great to propose that that India considers either renting some space or getting their own module attached to someplace like the orbital reef where they could conduct agricultural research.
In the interest of time, I have only identified 3 people, Femi, then Damu, then Shobhan. Go ahead first.
My name is Femi Anna Thomas. I'm working as project scientist at National Institute of Ocean Technology. Thank you so much for the platform. I wanted to ask, now we know that there is vast scope for microbiome research in India, and there are different, people working in different diversities on microbiome research. How can we sensitize the citizen as well as the stakeholders and the funding agencies to bring under one umbrella, to address this? What can we do as a citizen? Meaning what can we contribute as a citizen science about the microbiome research, about the importance of microbiome research?
Very good question. So all the students, for example, now you have seen several students from several things. You guys just collectively write a white paper, send it to me, I'll publish. Okay, I promise. So take Sastra University, take Indian Institute of Science, and you and National Institute of Oceanography, wherever. Okay, bring and ask this question, write a 1-page or 2-page white paper. I'll publish it right away. Damu, next.
Hi, good. I'm Damodaran. I'm a faculty from MGM Advanced Research Institute, Pondicherry. Actually, we are lacking in culturing unculturable bacteria. That is, culturomics is there, but it could solve the problem in some extent to culture uncultured bacteria. So we are lacking, not only in India, the whole world is lacking a co-culturing of all the species together, because the bacteria, fungi, virus, all together, they depend on one another. As Dr. Karthik presented, the metabolites produced by one bacteria can be taken by fungus or by the virus. The fungal can produce antiviral peptide that can kill the virus. So in a particular environment, if one species is killed, that will affect the microbiome diversity. For example, I'm working on human gut microbiome for the last 10 years. I'm trying to associate what is the gut microbiome diversity is affecting related to clinical outcome. Patient with cardiac disease can have difference in gut microbiome disease. So already there are biosensor available for identifying some particular bacterial space marker. For example, if you have methane, more methane in your oral, flow, sensor, already biosensor available from 2013 that based on a methane level in mouth, flow, you can identify whether methanogen is present or not. Like that, some bacteria produce propionic acid that can affect your neuron system. So India can focus on developing biosensor based on microbial markers.
Absolutely. That's what we talked about it in the beginning. Metabolomics is one area that India can move on, because DNA may be expensive, but metabolomics is not that expensive compared to that. Shobhan, then Pratyay.
I have a follow-up question of this.
Oh, go ahead.
Okay. So so this is Pratyay from IIT Madras. So I have a follow-up question. We usually use genes as a biomarker for cancers and all for past decades, long time. And people started using microbes as a biomarker. But there is a lot of diversity among us due to diet and environment. So how good it is or how robust it will be using microbes as a biomarker for?
I don't know, because microbes is so vast, and you don't have the bad microbes only. You have 99% of them good. Without that, you cannot even live, right? So, but thinking outside the box is always good. If some but you have therapy if you are going for a targeted thing and personalized medicine and all, then microbes can be used to used as a countermeasure. But I do not know, is that what you want to do, and then see the disease and microbes, or how do you use those things? But again, write a white paper, and then that will bring the awareness among the faculty, and they can think about it. I think. Sure. Because I'm not giving the phone to anybody, mainly because I'm rushing up to close the session here, because we have to catch the flight as well. Go ahead, Shobhan. Hello, everyone. After this, I'm going to give here everybody can take 1 minute to finish the thing. Go ahead.
So yeah, I'm Shobhan Karthik, undergrad at IIT Madras. So I have a follow-up on Indu's question. So how challenging is it going to be to make that database, the platform? Like you have to network with other scientists across India, and how
It
challenging
cost $40 million for him to get that NMDA, right? NMDC. So he was asking $100 million. So he got only 10 million first. Then after that, he got another 30 million. You have to come up, ask a big question, as he said before already. Think big, keep writing. So you have writers in the group also. Keep educating the people, as Fatih said. Keep educating the people. How do you educate? Your professor may not listen to you. You write open form. Nobody's going to stop you, open forum. So write that, okay? And when you're writing, you'll come to know whether anything happened or not. Because when you write, then somebody will say, hey, I have this. I have it. Then you started gathering the information. Same thing, okay, for you. Okay, with that, I'm not shutting up here, but there are some, okay. Very short question. No. He's going to give you the phone.
There was a question about the publications, the problem with publications. Just like with our Astronomical Society of India, we have a journal which is published together with Indian Academy of Sciences, Journal of Astronomy and Astrophysics. The same thing, it can be done with the Omics Society. Omics Society can have its own beautiful journal called Omics.
But it pays $4,000 right now. We have Omics Society, not one journal, multiple journals are there.
In India?
I don't know about India.
No, I'm talking about India.
You open it in Indian Institute of Technology and make it public, open access, no problem. We have in Indian Institute of Science also. If you want to do it, start from there. Every 3 months you can publish, hundreds of papers if you want. Free.
No, it has to be specifically Omics Society of India.
No,
that's what and it would have its own journal.
That's right. That's a good suggestion. We'll take a note of it and we'll write it down. And I'm going to pass on the mic from to Alex to start, and then final thoughts. Please don't go more than 1 minute. She's having the timer.
Okay, very short, very quick. Yeah. So my point is, I think you should have a kind of Indian Microbiome Society or Indian Microbiome Initiative. This is something that I'm trying to do now in Saudi Arabia. So it's building a microbiome, failing an initiative from the scratch. And they have some very good human resource and critical mass in clinical microbiology, clinical microbiome. What I'm trying to do now is connecting, the dots and some particular PIs with different expertise and fields. And we are writing now one document that via the South Microbiome Initiative, trying to leverage, trying to push hard this field, the microbiome science. Yeah. Hi. Well, yeah. As a fungi, as a mycologist, so I want to just comment this. So in microbiome studies, we need to think about fungi. We need not only consider bacteria as the main source of diversity there, because we know, as he said, the interactions between fungi and bacteria and viruses and other prokaryotes, it's important for multiple complex systems. So my thoughts are just think about fungi.
Okay, community of practice. I'm going to go back to what I said originally. I think this is a large network program that everybody's been talking about. Everybody has specific research interests, but I think it's really broader. You have to have a comms component. You have to have some expertise, from a internet perspective, and you have to have the research critical component. But with that, within that research component, you're also going to have multidisciplinary folks that you need to be involved, right? I mean, you have to have. You can do the good microbiome research, but if you don't have the engineer, for example, in the back end to help with the application and understanding how to communicate what that application is, that's going to be problematic. So I think it's going to have to be an integrated solution here.
Thank you. You heard from me a lot, so I will give it to him.
Just to kind of reiterate what I said before, that I think that culturomics can be a nice niche here. I think fundamentally all of it boils down to money. right? You need more money coming in. You need to get to convince people at the highest levels that there needs to be funding for this type of research, and that means demonstrating value. But in a kind of bottom way up, again, culturomics is missing in a lot of laboratories in the United States. It's also a labor-intensive and instrument-poor area, and that could be a nice niche to initiate kind of building strategy. You can use it as a way to collaborate with laboratories around the world, and then keep on bringing money in, at which point you'll be able to justify purchasing instruments and demonstrate to the government that it's got continued value. And then just last thing, in terms of the journal, there's a journal called PeerJ, which could be a good model. You can buy a lifetime subscription and then free publication, as long as everybody is on it, could be used the same way to make sure that it's open access.
How much is PeerJ?
Yeah, so you can buy a lifetime subscription. I can't remember, $1,000, and then I know, but what I'm saying, the model is that as long as everybody is a lifetime member, then every publication is free. So and they, but still that concept could be a nice way to get an open access, inexpensive journal.
I think that personally, the notion of the microbiome and association with plants is, I can tell you, being associated with the current research that's being done on the space station, is an emerging notion. For a long time, people were only interested in growing the plants as plants. They only cared about them as plants. But I think that for India and for you as a researching group, making the strong connection between the plant rhizosphere and the plants themselves, and connecting it to agriculture. I mean, the way that you get people in your government to give you money is if they see connection to something of value in your country. And I just think that the strong agricultural base of India is all the excuse, the justification that you need for why you should be pushing this research forward and why you should be supported.
India, I strongly believe that India needs a microbiome initiative. It's touching so many different aspects of the society: agriculture, astrobiology, health, and so many other areas. Most important, it's interdisciplinary, and it's not a microbiology society. The microbiome initiative should be interdisciplinary, and I think that's one of the major strengths as well of India. And I already see engineers and biologists, mathematicians interacting here. Funding, how much the funding should be? Ask how much is the funding for the Indian Space Agency? How much is the funding for NASA? Why microbiome initiative in the US shouldn't have an equivalent funding as NASA? Actually, this has been proposed. Microbiome, we are living in a microbiome planet, in a microbial planet, and we have not really explored it. It's touching the health of every being and every human. And we have not really explored it. We do not understand it yet. So the funding should be at the order of billion. So it's very important to make the government, the Indian government, understand that and organize pretty much the community around this initiative.
I'm going to make just a couple of points. So one thing is about the publications and so on that people are accessing. I think funding agencies are receptive. You just have to push them through, and also need institutional support. But we can discuss about this offline if anybody wants to do it, because I actually sort of sit on the committee for open access at IIT Madras. That's point number one. And point number two is, I think lots of initiatives are there. For example, we were talking about a database, having all of these projects that are going on in the country. In fact, that was sort of mandated by the Prime Minister in one of his inauguration speeches at CSIR and so on. So there is intent at the top, if you will, but it just needs a group of motivated people to follow it up. I think all of these are lagging behind because we do not have enough human resources constantly dabbling at this problem, right? We go on to other things, as Venkat was saying, and I think we need to make a concerted effort to identify some of these projects and really push them through over the next few months?
So I think that we are living in the exciting times, and I think there's tremendous potential for you as scientists, for you as human being. And there's a phrase that in France, which is popular, which is impossible is not French. And I'm going to change it to impossible is not Indian. And what I try to convey here is that everything is possible. And, you know, don't take no as an answer. I think you should be bold. I think from a scientific perspective, it's all about collaboration. It's all about interaction. It's all about networking. And I think there's tremendous potential in this country. And then I will finish with something which is says it's Latin. It says per aspera ad astra, which means that through hardships to the stars.
Thank you. Awesome. One last closing notes here. After that, Karthik Raman is going to do the next one. This is a tremendous effort that you guys have taken. Come here, and more importantly, very, 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 interactively, you have talked together here. I am so happy. Otherwise, you come and listen. We don't have even time for more than 5 minutes, and then you leave. So now, I'm so happy to organize this one, and the more things comes in the future. Okay, kudos to you, all of you guys here. So here is.
Just going to have a quick valedictory, and we usually used to hand out participation certificates and so on. Now it's all just going to be emailed, right? But I just have the duty of thanking a lot of people. Very nice slideshow going on behind you. So you can watch that and listen to me as I read out all of the folks I have to acknowledge. So I'd like to first start by acknowledging Dr. Prakash Arunachalam, who funded the IBSE initiative. Right? We have a $1 million endowment, which helps us organize events such as these without debating too much. Right? So thanks to him and the Office of the Dean of Alumni and Corporate Relations for enabling this. Also the Office of Global Engagement for funding this. They actually funded a third of the symposium. Through the IITM Institute of Eminence Initiative. So really grateful for that. And I think all of you know how much of work Ashmita has put into this. So a big round of applause for Ashmita. I think she's still sitting outside doing some organization. So, so really can't have her in here. And then I'd really like to thank all the volunteers who did a great job. Shobhan, thanks for the Slido particularly and everything else. Pratyay, Harshita, some of them are still running around. Sunanda, all round work. You have that right on the screen when she comes up. Right, so this was mostly her doing the decoration. Aishwarya, then Venkatesh, Srijit, Amrish, Sai, Amrita, Roshan, Rajiv, Dinesh, Vishwa, Brinda, Lavanya, Indu, Nilesh, Virendra, Yuvaram, and Martina. So thanks to all of these people. I think they really made a huge difference. I'm going to make them all watch the lectures on YouTube later on so that they catch up with all the science that they missed during the symposium. And I think that also goes for me. I'd like to really thank the speakers. Right, we had 15 in-person speakers and 10 virtual speakers, and many of the virtual speakers were very participative throughout. And I think that was really good. And Fatih and Nikos were helping us, something as simple as getting a DVD and bringing it here on time so that we could set it up. So I think it was everyone really, pitched in and contributed. Really amazing. So we had 30 male participants and 26 female participants. So I think the participant-wise, the ratio was fairly good. But I think speakers, we had quite a few cancellations, but we're very happy that we could have a few speakers, and we also had a childcare grant for one of them, so which enables them to travel. And I'd like to really thank, of course, Venkat for all the organization. I think he's been right from the get-go, setting this up, to conceiving the idea and getting all the people and handling all the cancellations, finding the appropriate replacements. It's just been fantastic. I think he's been the one thread that's held this entire thing together. So a big round of applause for Venkat. And yeah.
So actually, I have to thank Karthik, because without Karthik, nothing would have started. I just had an idea. He's engineered this to make it happen. So big round of applause to him.
Thank you. And also to Himanshu, he's been like really busy with so many other things, so he's not participated in his usual self in the symposium, but he's been, following up a lot of these things and sounding board for a lot of ideas and so on. And lots of folks at IIT Madras, I'd like to thank the Dean, Mahesh and Raghu, who are here for the inauguration. Then Lisa, who's been around all along, so a big round of applause for her. And then we have the folks at the audio. We have Vaidishwaran, Taufik, and Mohan Raj, who are with the audio, and Prabhu Raj doing the photography tirelessly, and I think Vignesh, right? Vignesh doing the photography and Prabhu Raj with the video. So thanks to both of them. And then Vinod and Rudra at the Office of Global Engagement, Bharati and Sri Priya at our NPTEL office, who've been handling the payments and so on. Sairam of the Media Cell, Robert Bosch Center, the guest house folks, lots of folks at the hostels who've managed everything. Devamita for helping out with all the certificates. And of course, the folks at the cultural program yesterday. I think you all really enjoyed it. Sri Varshini, Sunanda, Prahlad, Sri Vidya, for doing it all at like zero notice, basically. And also to Aishwarya for really a very nice comparing of the entire event. And Kirubai for getting the audio set up on time. And we had Shahnaaz helping with the flyer designs. Aditi, who's still working on the conference reporting. She'll be preparing our summary and white papers. And Kadir Travels, Karpaga Travels, Walker Associates, Print Up for printing, and Tejus for the mementos, and Nandini Sweets, Hemas, and Pixie Photos for photography, and Satya and Shubha at Ramada Plaza, and Eden for the hospitality and so on. We just, this event wouldn't have been possible without all of their help. So really grateful to all of them. And last but certainly not the least, all the participants for really making this a very energetic event, as Venkat was mentioning. And I'm not going to stop at that. And I hope we can continue interactions. I'll probably set up a Slack channel or something where we can continue to interact with all the speakers and amongst ourselves, right? I think we need to really push the Indian Microbiome Initiative forward, and this would be a good excuse to start it. Thank you. So we can now head out for lunch, and that's about the end of the symposium. We have a few other programs for the speakers are going to meet with the dean and so on, but I think this is officially the close of the symposium. So let's enjoy lunch and continue our discussions.